Hello Bio 1 or Bio 220 students, and welcome to this last lesson into Module 1. What we're going to cover into is actually, infectious agents and immunology, practical applications of it. And might I say, this is going to be one of the more fun lessons to give. Well, we're going to talk about vaccines, as well as differentiating the different types of vaccines, and talk about agents used within vaccines. Well, getting into types of vaccines, we're going to talk about a term called variolation, which is the inoculation of smallpox into the skin, or the application of smallpox onto the skin. It was a way that Jenner inoculated cowpox into cows, which helped prevent the future disease and fatal disease smallpox from killing cows. Essentially, vaccine, it's a suspension of organisms or parts of an organism to help produce immunity. So let's look at vaccine functions. Or I should say, the origin of the word vaccine. Essentially what vaccines are is they provoke a primary immune response, which causes our bodies to eventually form antibodies and memory cells to that infectious agent. Typically produces a rapid, intense secondary response. And the key is a concept called herd immunity, which is the immunity when a large proportion of a population gets vaccinated from a specific agent. Typically in areas where outbreaks are sporadic, if enough individuals become vaccinated, it helps a disease from moving to new people. This is just an example of the variety of diseases that vaccines are used to help prevent those diseases that at one time were very effective at ending life, but nowadays are more controllable. What you're going to see is that there are different types of vaccines, some that are attenuated, some that involve what's called a killed virus, and we're going to get into many different types. Here's actually the core lesson material. The different types of vaccines. Essentially, what is the vaccine composed of? If a vaccine is a live attenuated vaccine, it consists of a living, quote-unquote, living version of a virus, but it's a very weakened version of that virus or pathogen. And it closely mimics an actual infection so that our body forms a very strong immunity towards it. Now, inactivated or killed vaccines, basically they consist of just a virus that was either heated or exposed to chemicals that made it no longer functional and reproducible. And here's something about inactivated or killed vaccines. They're typically safer than live vaccines because even a live attenuated vaccine, it's still a living pathogen. So even if it's weakened, in the case of a weakened virus, it may eventually replicate and mutate into a virulent or infectious form again. Whereas killed-off vaccines, they're no longer pose the danger of actually becoming a virulent form. But typically these require additional booster shots to help the body know that this is something it does not want inside it. So influenza viruses, they're grown from embryonic eggs. Types of vaccines and their characteristics. A subunit vaccine involves antigenic fragments to stimulate an immune response. Now, types of subunit vaccines are recombinant vaccines, which include a subunit vaccine produced by a genetic modification. Virus-like particle, or VLPs, these resemble intact viruses but do not contain the genetic material. So essentially, it might contain a part of the outside protein coat, but not the actual infectious material inside the virus. And then there are also toxoid vaccines, which contain inactivated toxins. An example of antitoxins are, serums containing antibodies against a toxin. Typically, vaccines do not actually contain antibodies, but they contain either parts of the actual virus. The reason being, if the body is injected with something that should definitely not be there, it will develop an immune response to eliminate that substance. So there are also conjugated vaccines, which are used for diseases in children and people with poor immune response to capsular polysaccharides. And then there are nucleic acid DNA vaccines. These involve injecting an exposed string of DNA, which is then used to produce an antigen encoded in that DNA strand. Protein antigens carry to the red bone marrow, stimulate humoral and cellular immunity. So, development of new vaccines. Typically, it's less profitable than medicines because most vaccines, they're designed to give long-term immunity, or they're very specific to one type of infectious agent, whereas many medicines are good for a wide range of infectious agents. Typically, vaccines are developed without the use of animals. Many vaccines actually use plants as a source. And there's a goal to try to make oral vaccines that don't involve actually injection with a needle, but can be taken orally through a simple spray or nasally through the nose. There are developments to form vaccines against chronic diseases, and there is also reverse vaccinology for the development of cellular immunity, making our cells stronger. Now, many technologies include nanopatch, which delivers a dry formulation or a dry concoction of a vaccine to the skin. Again, it's getting past many people's fear of a injection with a needle. Essentially, the skin contains a high number of APCs, and, it requires no refrigeration, so it's a way of saving cost on preparing that vaccine. Also, multi-combination vaccines, which help provide vaccination against a multitude of infectious agents. That's the goal, to help make vaccine production more profitable. A very critical component of vaccines is an adjuvant. These are chemical additives, typically aluminum or alum, which are added to vaccines to improve effectiveness. Essentially, the infectious material of a vaccine or the mimic of an infectious material, say part of a virus, your body might notice it, but it will only react to it so much. An adjuvant really tells the body, this is something that you should pay attention to, and you should form a very strong immune response to whatever this infectious agent is. So essentially, adjuvants, they help inflame that area that was injected with your vaccine material to help your body form a much stronger reaction to whatever's in there. Basically, it helps your body know, hey, I didn't like what was in there, so now I'm going to form a lot more antibodies than I would have if it was just a little bit of the viral material. In the US, we only use alum and as an adjuvant to increase the effectiveness of vaccines. And essentially, by increasing inflammation, it increases the innate immune response. So the key of an adjuvant is it greatly increases the effectiveness of a vaccine by increasing our body's natural ability to distinguish when it doesn't want something in the body or when something shouldn't naturally be in the body. Now, as far as the safety of vaccines, there are occasions where vaccines may cause a disease. That's because if you're using a live or attenuated vaccine, if a attenuated virus that's very weak, it may actually replicate and mutate into a more virulent form. However, there has been a misconception that vaccines have been linked to autism, and there has been no medical or scientific research that has shown that vaccines might cause autism. More than likely, it's more likely a genetic cause that occurs at birth instead of something that is given to a child later in life, like a vaccine. Now, vaccines, they're actually a safe and effective means at preventing many extremely deadly diseases that would have killed older adults, children, many years ago. Okay, getting to some new terms. Sensitivity, which is the probability that a test is reactive if the specimen is actually there. And then specificity, which is the probability that a positive test will not be reactive if a specimen is definitely not there. Immunologic-based diagnostic tests. These involve interactions of the humoral antibodies with antigens. They're known as antibody. Known antibodies can identify an unknown pathogen if you know what antibodies are linked to that pathogen, and a known pathogen can help identify an unknown antibody. This is essentially performing diagnostic tests to see what you might have. You might use an antibody test to see what kind of pathogen you have if you're not sure what it is, or you might use a known pathogen to help determine was the function of an antibody if you haven't discovered what that antibody is used for yet. Monoclonal antibodies. One example is hybridoma, or immortal cancerous B cells, myeloma, were combined with an antibody-producing normal B cell. Hybridoma, they produce monoclonal antibodies. So you can use those to determine if you have hybridoma. mAbs, well, they're uniform and highly specific and are produced in large quantities, which is why a diagnostic test is very effective at distinguishing if you have hybridoma. Now there are also chimeric monoclonal antibodies, or mAbs with a mouse variable region and a human constant region. Essentially, you're taking different regions of an antibody from different agents. There are also humanized antibodies, which are mAbs that are mostly from human, except for possibly a mouse antigen binding site. And then fully human antibodies, which are antibodies produced solely from a human gene that was transferred to a mouse. So the production of monoclonal antibodies can involve the injection of an antigen into, say, a mouse, which then goes into its spleen, and, well, through a process of cell division, can eventually be used to produce more of that antigen, or to test to see if there's a presence, I should say, of cells that are actually hybridomas, cancerous cells combined with healthy cells. So, serum studies, which involve studying blood. An example is studying the blood of an infected cow, which would have a considerable amount of antibodies against a certain infectious pathogen in his blood. Essentially, if a organism has already been infected with something or is currently infected with something, it will naturally form antibodies. And through a serological test or a blood test, a researcher can determine what antibodies are present and what levels of those antibodies are present. And there are precipitation reactions, which are reaction of soluble antigens with antibodies to form large interlocking aggregates called lattices. An antigen-antibody combination forms, followed by the formation of this lattice that precipitates or crashes out of solution and forms a solid, so it's no longer dissolved. There is a precipitin ring test, which is a cloud line forms where there is an optimal ratio of antigen-antibody, and we'll look at what that is. Essentially a precipitation curve. It's a way of determining when you have a good amount of not too many antigen and not too many antibodies, but a good combination or mix of both. And to look at it even further, essentially what we're looking at here is the precipitin ring test, where essentially you have a solution where the antigens are soluble on one side and the antibodies are present in another. And then in the middle, you'll have a precipitation, where essentially the combination of antigen and antibody is no longer soluble in that solution. Therefore, it forms a solid. Or a precipitation band. Precipitation reactions, one of which is an immunodiffusion test. It's a precipitation reaction carried out in the agar gel medium. Precipitate develops at the point where the optimal antibody-antigen ratio occurs, and immunoelectrophoresis combines electrophoresis with immunodiffusion. It helps separate proteins from human serum by helping remove it from the overall blood. Now, agglutination reactions. These involve particulate antigens binding to antibodies to form aggregates or coagulation. Direct agglutination tests. These detect antibodies against large cellular antigens, and well, they help measure a concentration of antibody in a serum, which is known as a titer. And a rise in titer or a rise in concentration indicates that a greater immunity to disease was formed because more antibodies were produced. Essentially, that's why adjuvants are added to vaccines, so that a greater titer is produced in the end. Now there is seroconversion. It's a significant change in titer as the disease progresses, say from not having much antibodies to having a significantly greater amount or a greater titer. And agglutination reaction. Well, basically immunoglobulins or immunoglobulin M essentially binding to receptors on infectious bacteria, forming aggregates. A process of measuring, titer with direct agglutination test. Well, Read. This is just a way of, let's see. Essentially, each well in the microtiter plate contains, from left to right, only half the concentration of serum that is contained in the preceding well. Each well contains the same concentration of particulate antigens, in the instance, red blood cells. Now what happens in the side view is the agglutinated. In a positive or agglutinated reaction, sufficient antibodies are present in the serum to link the antigens together, forming a mat or a complex of antigen-antibody on the bottom of the well. Non-agglutinated situations. In a negative reaction or a non-agglutinated reaction, not enough antibodies are present to cause the linking antigens to particulate antigens roll down the sloping sides of the well, forming a pellet at the bottom. In this example, the antibody titer is 160 because the well with a 1 to 160 concentration dilution is the most dilute concentration that produces a positive reaction. So at what level is something observable? Agglutination reactions can involve an indirect or passive agglutination test where antibodies react with the soluble antigen adhering to particles and vice versa. There's also hemagglutination, which is the agglutination of red blood cells surfaces, or the surface antigens on red blood cells, to complementary antibodies. It's used in blood typing, determining if you're a blood type A, B, C, or D, or blood type A, B, or O, as well as are you positive or negative for a rhesus factor. Now, viral hemagglutination occurs when viruses agglutinate red blood cells without an antigen-antibody reaction, and examples of those viruses could be mumps, measles, as well as influenza. Just an example of an immune molecule reacting with, say, antigens on a latex bead or antibodies reacting to antigens on those beads. Well, reaction in a positive indirect test for antibodies is what is shown on the left. When particles are coated with antigens, agglutination indicates the presence of antibodies, such as immunoglobulin M, shown here. So essentially, some source with antigens is inserted to see if there's any antibodies or a specific type of antibody. And if a complex forms, then you know that you have that particular antibody. In a different sense, a reaction in a positive indirect test for antigens, which is shown on the right, is when particles are coated with a monoclonal antibody, and agglutination would indicate the presence of the antigens after they bound to those antibodies that you know are there. Now, viral hemagglutination. This involves red blood cells and viruses interacting to form hemagglutination. Neutralization reactions. Essentially an antigen-antibody reaction where the harmful effects of an exotoxin or a virus are blocked by antibodies to the toxin, called the antitoxin. Viral hemagglutination inhibition test is used for subtyping viruses. Viruses and red blood cells are mixed with the patient's serum. If the serum contains antibodies to a virus, they neutralize the virus and inhibit hemagglutination. So complement-fixation reactions. Well, they're going to be when a complement's serum protein binds to and is fixed to the antigen-antibody complex. It helps detect when there is only small amounts of antibody present, and it also works for antibodies that do not work with precipitation or agglutination reactions. So it's one other way of determining the presence of antibodies. Fluorescent antibody techniques. Well, this is when you're going to involve a fluorescent dye to determine the presence of antibodies, or you're going to combine fluorescent dyes with antibodies. In a direct fluorescent antibody test, it's used to identify a microorganism in a clinical specimen. Whereas as indirect fluorescent antibody techniques, they detect a specific antibody in a serum. For example, anti-human immune serum globulin, anti-His-G, is added and will react with any antibody in the serum if it is positive or present. So, fluorescence activated cell sorters, or FACS. These can involve laser beams which strike a droplet containing a cell, or it can be a detector, where a detector determines the size and fluorescence of surface molecules, or imparts a charge to the cell, helping separate cells. A nice technique, which is a direct ELISA, or enzyme-linked immunosorbent assay. Direct ELISAs, they help detect antigens, and well, it involves a sample containing antigens mixed with an antibody. Enzyme-linked antibodies react with the antigen, and they are then detected by adding a substrate to the linked enzyme. Usually a color is produced in the process. But another type of ELISA assay is a indirect ELISA, which is actually used to detect antibodies instead of antigens. One example is Western blotting, which identifies proteins via electrophoresis and a blotter. Electrophoresis is just a way of separating components by applying an electric current, and then a blotter is just a way of putting a sample through a gel to help separate different components, either by charge or by how much that component weighs. Or the size, I should say. Now, monoclonal antibodies can be used in a pregnancy test to determine if, say, the child has a different blood type than the mother. Well, these can help determine if other medication is needed so that no dangerous situations occur, or to determine if someone is even pregnant or not. Future of diagnostic and therapeutic immunology. Well, these tests require less human judgment and personnel or people. They involve polymerase chain reaction and deoxyribonucleic acid probes, so PCR and DNA probes. They're very inexpensive, and they're simple tests for developing countries, and they help prevent disease prevention and therapy. Essentially, PCR is really good at amplifying a small amount of something to where it's large enough to either be observed or to make enough of it to be meaningful for, say, a medicine. And well, that concludes chapter 18 of this first living module. Well, to be honest, you all have a lot of material, and you definitely have my respect for taking an online microbiology course. Anyways, well, peace, and see you in the next learning module.